So basically, translocation has to happen first. So translocation is the transfer of tRNA from one binding site to another. Okay, so let's just say that our tRNAs that carry the methionine and the glutamic acid here, okay, they have been translocated towards the E site once they have been used. Okay, so they are now considered as a free tRNA. That's why they move out of the ribosome here in the E site or the exit site. So now new tRNA is going to attach onto the A site, okay, and then the tRNA on the A site is going to translocate onto the P site. Okay, so when that happens, whenever the two amino acids are adjacent to each other, there is a formation of the peptide bond. Okay, so when more and more amino acids are being condensed together to form the polypeptide chain, then that is a process of elongation. Okay, so the next process that is going to be discussed next is the process of termination. So the last process of translation is known as the process of termination. So termination occurs when we have already formed our polypeptide chain and when our ribosome here recognizes the presence of the stop codon. So the stop codon is actually made up of nitrogenous bases such as U, G, and A. Okay? So when the ribosome has recognized the presence of the stop codon, no tRNA is going to attach onto the A site, and therefore the last tRNA that is present on the A site will be translocated on the P site, and then from there, upon the recognition of the stop codon, this releases the polypeptide chain away, and then this tRNA that is present in the P site will be translocated onto the E site and out of the ribosome. So once we have already formed our polypeptide chain, from there it will decide what function it will serve, depending on what amino acid that has been condensed altogether. Okay, so for our starting points, then actually the next process or the next part that is actually included under nucleic acids and proteins is the topic proteins. So this is already being learned by HO students, and they are actually required to know the structure of amino acids. So they have learned this in the previous topic, such as in the chemistry of life, and they're supposed to memorize the structure. So I have drawn here two amino acids. Okay, so for our starting points in the topic proteins, they should know the structure of dipeptide and how a dipeptide is being formed. So I have drawn here two amino acids that are adjacent to each other. So a dipeptide is formed, okay, because of the interactions between the hydroxyl group of the carboxyl part here and the hydrogen from the amino group in here. Okay, the student should be able to know the parts in an amino acid. So because of the interactions between the hydroxyl group and the hydrogen of the amino group, okay, and they undergo the process of condensation reaction, meaning a bigger molecule is going to be built by removing a water molecule. So we are able to remove a water molecule here. As a result, we are able to form a bond that is going to be present in between the structures, the carbon double bonded to O and nitrogen double bonded to H. So the bond that is present in between them is known as the peptide linkage. Okay, so the student should be able to know that the most basic linkage is present in amino acids is the peptide linkage. So next, we are going to discuss about the levels of protein structure. So the next area that is needed to be discussed under the subtopic proteins is the levels of protein structure. So it's going to be helpful for students if we are going to provide diagrams for each levels of the protein structure. So we can start with saying that there are 4 levels of protein structures, namely primary, secondary, quaternary, tertiary, and then quaternary structure. So we are able to, or students should have an idea of what the levels of protein structure are if we are only if we are only able to concentrate on the components of the protein structure and the linkage or the bonds that is able to be formed within the protein structure. So in the case of primary structure, it is composed of an amino acid chain or a polypeptide that is going to be linked all together using a peptide linkage. So previously, we have discussed that for us to be able to form a dipeptide structure, there should be the interaction between the hydroxyl group of the first amino acid and the hydrogen group or hydrogen of the second amino acid, and they're going to undergo the process of condensation reaction. So as a result, peptide linkage is going to be formed in between the 2 amino acids. So if we are going to condense all amino acids together, just like what we have discussed in the process of translation, we are able to form our polypeptide structure. So the next one is the secondary structure. So this time, the secondary structure is a more complex structure compared to primary, wherein it is made up of a polypeptide formed into a specific structure. So whenever the polypeptide is assuming a specific structure, namely the alpha helix structure of beta sheets, that is considered as a secondary structure. So whenever the polypeptide is going to be coiled into a chain, therefore that structure is known as the alpha helix. An alpha helix structure will only be formed if there is an interaction between the amino acid residues at specific points along the length of the polypeptide. So as a result, there is a hydrogen bond that is going to be formed within those points. So on the other hand, beta sheets will only be formed if there is an interaction between the amino acid residues between 2 amino acid polypeptides, between polypeptides that are parallel to each other. So as a result, if there is a conformation or there is an interaction, that is going to be formed in between these 2 parallel polypeptides, then it will form a hydrogen bond. So as a result, the beta sheets is going to conform a structure of being folded, whereas for alpha helix, it is going to be coiled. Okay, so the third level of the primary structure is the tertiary structure, wherein whenever a polypeptide is going to be further folded into a more precise and compact structure, then it's considered as a tertiary structure. We can also point out to our students that tertiary structure is only present whenever the polypeptide is going to be in 3D structure. Okay, because the difference between secondary and tertiary structure is that secondary structure often comes in 2D structure only, whereas for tertiary comes in a more 3D structure. So comparing the tertiary structure with the secondary, tertiary structure has more chemical bondings that is being formed within the polypeptides. So it's actually capable of producing 4 different types of chemical bonds, namely hydrogen bonds, ionic bonds, disulfide bridges, and van der Waals forces. Actually, the students don't need to go into deeper detail on the areas wherein, like, which groups within the amino acids that are going to be forming these types of chemical bonds. So just knowing the 4 different types, I think that will be sufficient. So the last level or the last structure that we have is the quaternary structure. So the quaternary structure is actually composed of two or more polypeptides that are going to be formed all together to form a single protein. So a usual example of a quaternary structure is the hemoglobin. So the hemoglobin is actually a conjugated protein, meaning when we conform two or more proteins together. In this case, hemoglobin is made up of 4 polypeptide chains that is actually attached all together with a prosthetic group. A prosthetic group is a non-protein group. So in this case, the prosthetic group that is present in hemoglobin is the heme group. So the heme group contains the iron, which allows the hemoglobin structure as an efficient protein molecule that is capable of transporting oxygen all throughout the body. So if the students would be able to give an example of each primary structure, which is actually it's only the quaternary structure and the tertiary structure that we are able to give an example of. So later on, we are going to discuss more of the fibrous and the globular proteins. So another area that is actually being discussed under the subtopic proteins is about the fibrous and globular proteins. So one of the most common questions that is going to be asked in IB regarding this area is to differentiate the two structures. So by knowing the differences between the two structures, then we are able to give or provide more marks for the students. So I suggest that I always tell my students that if there is a question regarding comparison or like asking a difference between two structures that are fairly same but not really the same, but anyway, if it's going to be a comparison question, then I usually tell them to make a matrix or provide a table. So in that case, we are actually systematically writing down the differences between the two structures. So in this case, the difference between the fibrous and the globular proteins is, of course, their structure. So in the case of fibrous proteins, they are more of elongated and coiled, whereas for globular structures, they are more spherical or rounded, and they're more compact. So we can also add that fibrous proteins, they are made up of basically more of an alpha helix structure. And alpha helix structure is more of like a secondary protein. So, for example, if we are going to put together alpha helix structures, then it is able to form into a more structural protein. So, usually fibrous proteins are insoluble with water. And an example of that is the keratin and the collagen that is found in our hair and in the skin. So in the case of globular proteins, since they're spherical and they're compact, they're actually made up of two proteins. They're made up of alpha helix structure and the beta sheets. So if they are being formed all together, they actually conform into a more compacted or compact structure. So in this case, globular protein as compared to the fibrous protein, they are soluble with water. So in this case, globular proteins are usually being used in chemical reactions, and they are usually they come into contact with water. So that's why their usual roles of globular proteins is being used for being used as enzymes or hormones. So these are the most common differences between fibrous and globular.